Welcome, everybody. Welcome also all those online. It's my great pleasure to host this seminar that is jointly organized by the AMR Center, the Antimicrobial Research Center, as well as the McGill Structural Biology Center, better known as the CRBS. As is the custom at McGill, I would like, hopefully, this slide will now. McGill is on land which has long served as a site of meetings and exchanges amongst indigenous people, including, I'm really bad with words like this, but it's a great place where meetings have always held, and McGill is continuing this tradition, and we're very happy and thankful that we can host our speaker here on this territory. Which arrow was I allowed to push? There we go. So this is our speaker of today, Robert Bonomo. I've been saying this, his last name is wrong for many years, so you don't know that, but that's okay. Robert comes originally from the Bronx, and he has a very interesting and long career. I think I should highlight a few highlights. His CV is very long. I threatened to go through it in great detail because it's worth doing that. But he actually started out in a liberal arts college with a name called Hamilton College, which all of us should now know thanks to this wonderful musical, because it was named after Alexander Hamilton. He actually did very well over there, as he received an award for the current prize in classical languages. So clearly he moved on from classical languages, and after his foray in arts, he did go to medical school. He started out, he did this in Cleveland, and he actually has never really left Cleveland since then. He's currently a staff physician at the Veterans Affairs Hospital, the Louis Stokes Veterans Affairs Medical Center. Over the years, he has done that for many years. He's actually climbed up in the ranks. He's now director of VISN 10, which I figured out that is actually quite a big area of Veterans Affairs Hospital, where he's in charge of the geriatric research, education, and clinical centers. And he is also the national director and coordinating center of SHIELD, which is not the thing you know from Marvel. It's something else. It's a scientific and health initiative to combat emerging and lethal infectious diseases. Now, this would be enough for anybody's career to already do that. But this was just, I would not even say 50% of what he's been doing. In the meantime, he has also developed an academic career. And currently, he's also professor at Western Case University, Reserve University. He is professor in the departments of medicine, molecular biology and microbiology, biochemistry, pharmacology, and just let's add on also proteomics and bioinformatics just for fun. So it's not a surprise he has received many awards, in addition to his current prize in classical languages. I again will not mention all of them. I will just highlight two of them. He's a distinguished university professor, which says quite a lot, says a lot of things. He is also a fellow of the International Society of Antimicrobial Chemotherapy. And those who have seen his CV or looked him up on Google Scholar will understand that he is indeed in the top 1% of cited papers in biomedicine for I don't know how many years in a row now. He's published over 700 papers, citations. It's just embarrassing. I will not go there. I'm just thrilled that he said yes when I asked him to come here. So Robert, thank you for coming here. Thank you, Robert.
That was a very, very nice introduction. You know, it's humbling to hear things like that. And, you know, any successes that ever came to me was because of my wife, my family, and my great friends. So this is really more about them. So today I'm going to talk about a topic that I've been, like I told the students this year, I've been investigating since about 1990, beta-lactamase inhibitors. And it takes a village. thank you, Albert, for this invitation. But, on the way here, so many people at the NIH, the VA, the folks in my lab, I can't do an experiment. They did it all. And, they have been so helpful along the way. And I've also been very fortunate to receive the sponsorship from a variety of industrial sponsors over the years, which I'm very grateful for the research they supported in my lab. And I'm also grateful for the Antimicrobial Resistance Center. I mean, this is a great place, and I've had the privilege and honor to meet so many of you today. And your passion for your research, your commitment to improving the health of our world is impressive. And I tried to figure out what this molecule was, but we could talk about it later. I couldn't think of nicer words to put on a website. We strive to foster collaborations, contribute to local and global leadership, and enhance training opportunities. And I think of the missions of a university and the missions of a center dedicated to antimicrobial resistance. I think those are the most important things that you can say. So thank you for inviting me, and I want to encourage you to continue on your great quest forward. So antimicrobial resistance. Well, this is a very old slide. It wasn't even published in a scientific journal. But I used to have all these slides on the impact of antimicrobial resistance, but I decided never to use them anymore. Just one simple fact that by the year 2050, antimicrobial resistance will be responsible for 300 million deaths. That's all you really need to say. It's going to cost a lot of money, but you can't buy back life. And antimicrobial resistance as a cause for these unfortunate occurrences are really very, very tragic. Now, my lab has had this, quote, love affair with beta-lactamases. And, when you look at the field of antimicrobial resistance among gram-negative bacteria, beta-lactam resistance stands out. And, it stands out because bacteria have beta-lactamase enzymes, and they also have other penicillin targets, penicillin binding proteins, et cetera. But the beta-lactamases stand unique in the bacterial world for enzymes that are really, really good at breaking down beta-lactam antibiotics. And beta-lactamases had there's 4 members, 4 classes, class A, B, C, and D. The enzymologists were really smart when they did that nomenclature. They only knew 4 letters. So A, B, C, D was what they did. And class B is a little different than class A and class C and D because class B uses 2 metal ions. And when you think about that, you say, oh, they must be derived from different sources. And they are. But bacteria used these enzymes for the same common purpose. And when you look at the mechanisms of reaction to break down penicillin, the metal ions and the catalytic serine work the same way. They work the same way. And to overcome them, I tell everybody in the lab, if you want to, overcome beta-lactamases, it's not enough just to describe their epidemiology, but you have to fundamentally know the mechanism. And I think the point is working. Here is a serine-based mechanism. Here is a metal-based mechanism using 2 zinc ions, and the product is the same. You get this inactive molecule, and we could get into the details of proton transfer and electron transfer and how these different amino acids work together, but that's going to bore everybody to death. What everybody really needs to know is the fact that you have these bacterial elegant proteins that can almost defy our best synthetic efforts. And as long as we've been making beta-lactam compounds, we've been challenged in the clinic with the emergence of beta-lactamase inhibitors. And these beta-lactamases can be overcome by using specific inhibitors, and we'll talk all about that. But the mechanism by which these beta-lactamase inhibitors really is extremely important because unlike a beta-lactam compound that gets in there and gets turned over, the beta-lactamase inhibitors have multiple personae. They form an acyl enzyme complex, they form tautomers, they go back and forth between different intermediates because the compounds that are in there drive a different sort of chemistry. And what I always tell fellows in the lab and graduate students is that if we study the beta-lactamase inhibitors live and die at the bench, everything we do biochemically to characterize these enzymes is going to be anticipated in the clinic. So it's very, very important to continue to support that sort of basic research. The inhibitors in the pipeline, we all know ceftaz-avi, ceftolozane tazo, Imirel, Meroveber, and one that just crossed the finish line, sulbactam, darolobactam. We have some on the very near horizon, cefepime, taniborbactam, cefepime, and metazobactam. And metazobactam with cefepime just crossed over the finish line and is now a compound. We have some other compounds in development, cefepime zidibactam, a venatorex compound that's going to be met with an oral partner. We have other, cefpodoxime is going to be met with an oral partner that's being developed by, at that time, Entasis. There's a compound that's also being developed out of Wockhardt, ertapenem zidibactam, which is not the same as cefepime zidibactam. You're taking a carbapenem with a different biochemistry, essentially, than cefepime. And then we have some other compounds that are being married with some beta-lactams like ceruloborbactam and cefiderocol, and then a class by Cef is the avibactam timolopolol. So when you look at this, and I had the privilege of sitting on many study sections, and I've had the privilege of talking to many administrators in high academic places. When you show them a slide like this, is that they tell you, well, do we really need another beta-lactamase inhibitor combination on the market? And why should we be worried about the inhibitor pipeline when we have so many of these drugs on coming down the pike? Well, the fact of the matter is antimicrobial resistance is not getting better. And this one paper that was published in the Lancet, the global burden of bacterial antimicrobial resistance in the year, it wasn't public, it was came out a couple of years later, but in 2019. And this was supposed to be a comprehensive estimate of all the bad bugs all over the place. But, I read these things differently. This was really a paper on the increasing problem of beta-lactamase mediated resistance. So let's look at what is the real bad news. Well, the real bad news is not so much what's going on in North America. The real bad news is in sub-Saharan Africa, where you have a mortality of 27.3 deaths per 100,000. We are not paying attention to that part of the world and the struggles that they're going under. And then when you look at the demographics, everything that's in the bad zone, Western Sub-Saharan Africa, Central Sub-Saharan Africa, Southern they lead the pack in terms of the impact on antimicrobial resistance. And those are the most challenged areas in the world. So this is not only a scientific problem, but as Albert has probably figured out by now, I'm a little bit of a liberal, all right, but this is also a political, economic, social, and medical statement. So this is not just a disease process. This is something we really need to do something about. And the major infections, lower respiratory tract, bloodstream infection, and intra-abdominal infections predominate. But the resistance pattern in these is really, really much more than all the other types of infections. And all you have to do is go into a hospital medical intensive care unit and look at a patient who's having a lower respiratory tract infection, a ventilator-associated pneumonia. 5 out of 10 times, you'll probably recover a Pseudomonas aeruginosa, and 3 out of those 5 times, that Pseudomonas is going to be difficult to treat. But the 6 leading pathogens associated with that, 4 of them are gram-negative bacteria that are characterized by resistance: E. coli, Klebsiella, Acinetobacter, and Pseudomonas aeruginosa. So we have defined what the problem is nationally, and this impacts us because 3rd-generation cephalosporin-resistant E. coli. The big problem, a woman comes into the emergency department, fever, flank pain, you diagnose a pyelonephritis. What antibiotic do you give when the community prevalence of ESBLs is somewhere around 20%? Do you give ceftriaxone, or do you have to escalate up? Do you have to go away from a beta-lactam class of drugs and go for a quinolone? these are complicated decisions because even giving a quinolone is not an easy decision to make because of some of the concerns regarding some of the side effects of quinolone. So, all kinds of guidelines come out, and and we could talk about those guidelines over a cool, refreshing beverage. But the problem with these gram-negative bacteria are predominant, and I think a real game changer occurred when the carbapenem-resistant gram-negative bacteria came on the scene and really caused us to pause as we decide about empiric therapy. But it ain't cheap. In 2017 estimate, antibiotics are developing an antibiotic cost 1.5 billion, right? With the average sales is roughly 46 million per year. I know now that some people have said that the average annual sales of ceftazidime avibactam is around 300 million per year. So it's a little bit more than 46 million per year. But think about the environment, think about the milieu. You bring a new drug out in the market, and the first thing a hospital tries to do is to limit its use. So you're in a situation where you can't use an antibiotic. The company may have a different opinion about that, but they're also interested in the stewardship issues because they want the antibiotic to be preserved in the clinic, so they don't want it to be used willy-nilly. But they do have to recover their cost of investment. The return on investment is high, and for a drug that has a $300 million a year revenue, if it costs 1.5 billion to make it, I'm spending the first 5 years of that drug trying to recoup the money it costs me to do that. And then not counting the money it costs me to keep making that antibiotic during that time. So you can see that, we need another paradigm here. We need another paradigm. I like the word that was used in Nature. Antibiotic market needs to be reimagined. I can't think of a better, colorful way to say that we need a new paradigm. So why am I worried about this? Because the drugs like Tazavi, imipenem relum, meropenem vaborbactam, ceftolozane tazobactam, and cefiderocol, the new kids on the block, resistance is already emerging, and we need to take care of that. This is the established timeline. And, we were very fortunate in, we were invited to write an editorial for CID. And in the editorial for CID, I went bananas. For the first time, I said, well, I'm the first author, so I'm going to say whatever I want. And so I took an approach, and we just reviewed a case of this very unfortunate patient who developed some serious infections, all related to a line, and resistance had emerged, and they had a metallo-beta-lactamase producing strain, and then they used ceftazidime avibactam with aztreonam, which is an experimental combination, which may have been the right thing to do. And unfortunately, what happened is that the bacteria that was isolated from that patient was also resistant to that triple combination. So what's going on, guys? And when you go back, and I even this morning I was on with folks in my lab. We were looking at a collection we had from Century. People know that trial. Century trial was, I think, 2004. And we did some sequencing on those isolates. And even in 2004, we had the exact same phenotype that this kid had succumbed to because of that. So the bacteria are paying it forward, right? The old paradigm was, you introduce methicillin in 1960, you develop methicillin resistance in 1962. But then the game has changed, right? Imipenem is and ceftazidime reduced in 83. It took just a half a year for ceftazidime resistant Enterobacteriaceae to emerge. And now you look at what's in the pipeline. In 2005, our lab did a survey study, and we found that there was imipenem relibactam resistance, and imirel was introduced in 2019. 14 years ahead of that, we knew that. So the game has changed. The paradigm in antimicrobial resistance is that, and what I tried to say in that editorial, I know I'm trying to be personal, but, after all these years in the research, you take it personally, is that we're failing here because we're not anticipating and we're not responding timely. We have to anticipate the emergence of resistance. We need to develop our science so that when resistance does emerge in the clinic, we've thought about it in the laboratory. So TAZ-AVI, the new gold standard. And everybody, some of the people say, well, avibactam is a non-beta-lactam antibiotic. And I know, Albert, I'm not a chemist, but when I look at this part of the molecule and I look at this part of the molecule, they look awfully alike. So anyway, it's not a beta-lactam. It's a cephin bicyclic ring system. You can be special about it, but it's got the same warhead. It's the same thing. And, this was amazing. I remember when Sarah draws it, I, looked at some of the data with Christina Papp-Wallace. You have a organism with a KPC2 to ceftazidime MIC of 512, and you treat it with TAZ-AVI, and the MIC goes down to 0.25 to 1. If I developed that drug, I'd be in the pub that night. That's an incredibly potent advancement in clinical microbiology. And you have a OXA48, you go from 4 to down to 0.008. It's really extremely very, very potent because of the AVI inhibition, and also ceftazidime is not a good substrate for OXA48, but we don't want to go there. Too much biochemistry. And, but it's but there are some problems. It didn't do, TAZ-AVI didn't do well against the metalloenzymes. It couldn't bring it down. It had a little bit less activity against some enzymes that are common in the Middle East and in Latin America. And it did terrible against some others. So, it was not a panacea, but a real welcome. And I really enjoyed my relationship with, I really enjoy my relationship with Dr. Folko Vandenberg. He's a brilliant crystallographer, and he helped us understand how avibactam works. And, what I like to do is that, when we see this sort of structure, if you're a clinician, you're going to go, oh my God, I can't take that, all those bonds and lines, and, I'm going to walk away from this one. It's not worth looking at. But it's actually really simple. Because if you know that avibactam forms a acyl enzyme intermediate, you can follow that oxygen, and so that's where the next one goes. Then you can see that that ring system here. Then you could follow it back, and you can follow the sulfate. And by golly, I'm a crystallographer. I just learned everything I needed to know about crystal structures. I just look at it, and I'm not trying to be facetious, but sometimes there's a lot of truth in crystal structures. We just got to take the time, to pause and just follow the lines, and use the talents that God gave you, because she was a lot smarter than you were. Anyway, so in terms of real world applications, this was a nice study we had done with David Van Dyne, looking at colistin versus TAZ-AVI against carbapenem resistance, and it was an observational study, but the conclusion was potent. If your in-hospital mortality, if you use TAZ-AVI, was 9%. If you didn't use TAZ-AVI and you used colistin, your in-hospital mortality was 32%. You don't have to be a statistician to figure out that that's a significant p-value. And when you graph it out, it's, this is the hospital that's in the colistin group. This is the hospital that's in the TAZ-AVI group. This is the number of people that got discharged home who got TAZ-AVI. Not as many got discharged home when they got colistin, which even if they mixed colistin with something else, one good drug, TAZ-AVI, is better than two bad ones, colistin and whatever else you want to combine it with. So we learned a big lesson. And for the clinicians, if they're listening in, there's been a lot of really important studies that have been done. And I really like the name, Reclaim 1, Reclaim 2, Recapture, Reprise, and Reprove. So it's like resurrecting ceftazidime, saving ceftazidime. It's like saving Private Ryan. Anyway, these all showed that TAZ-AVI, did very well. Some of them were retrospective case series. But in terms of some of the big infections, particularly in Reprove, that against nosocomial pneumonia did well. But, they ain't no free lunch in beta-lactamase mediated resistance. And 11 years ago, we did some mutagenesis on the KPC enzyme. And, I had learned about how important mutagenesis was from studying SHV. So there's, okay, well, let's do something in KPC. So I knew that in SHV, if you made a mutation in the omega loop, you would get ceftazidime resistance. So we made a mutation in KPC at the 164 position, and we got ceftazidime, we got increased ceftazidime resistance. We made, and one investigator in Switzerland sent me a personal email and a letter to the editor, how he thought I was really out of line. I shouldn't be looking at these resistance enzymes. But my response was, you got to, if you don't study this, you won't know that this will occur. And then we did some more site-directed mutagenesis. Now it's a little tricky because you could interpret what we did as gain of function, and you need special limitations. You need to know what the hell you're doing when you're doing like that. And so we did quite a few investigations, but then the blank hit the fan when there was a bunch of Taz-Avi resistant patients that were found in the Pittsburgh, at the University of Pittsburgh, that had mutations on the other side of the MAGO loop, but functionally it was in that same area of the enzyme. So the bench research sort of anticipated what was going to happen eventually in the clinic. And we knew that the 179 position, that when you changed it, you did get increased Taz-Avi resistance, but we didn't realize it was going to be as clinically important as that. And then I had a different slide here, but I became obsessed, with trying to figure out this phenotype. And, we had a collaboration with Dr. Jim Spencer, Chris Schofield, and really brilliant. She was a graduate student at the time, Kathy Tukey. She did some really nice work. We did some further work with Foucault Vandenacker and his pre-doctoral student at that time, and we also did some of our own work with NMR. And I'll show you just a collage of what this was. And I literally became crazy. Because we looked at protein expression with those. We looked at RT-PCR, looking for mRNA reasons. We did differential scanning fluorimetry. We did NMR. We did more molecular modeling. And finally, when we even did mass spec, we found that, avibactam was not getting on as fast. We couldn't measure the AI, the Ki to form a complex, but, there was a problem with avibactam getting on the KPC variants. But the bigger problem was the ceftazidime. And this is why when you beta-lactam, beta-lactamase inhibitor combination, if you guys ever think of doing it, you got to think about this as a marriage. You got to have the right partners there, because if you don't have a good partner when you're married, it ain't going to be a happy relationship. So ceftazidime could have been rethought maybe, because of its property, but the KPC enzyme, can accommodate ceftazidime because, as Foucault demonstrated very nicely, being unable to map the residues in that region as a result of this one mutation, there was significant amount of disorder in the structural, in the omega loop of the enzyme, significant amount of disorder. And if you don't think that that's an important site in KPC, look at all the variants, KPC 31, 33, whatnot. All these KPC variants have emerged with a mutation at that 179 position, and they all have an impact. They all have some impact on ceftazidime avibactam resistance. And, looking at this from another perspective, Foucault nicely predicted that it was all conformational changes that allowed ceftazidime to be accommodated with steric hindrance, and that was part of it, and allowed the deacylation water to get into the active site. But we had to go back. We had to go back to some, it's really amazing when you look in the literature. It's really true. Some people were really smart years and years ago. We just forgot that they published, these really good things, and we failed to look, in PubMed. And I was talking about this with Professor Shahryar Mobasheri at Notre Dame, who's a brilliant chemist, and I was describing what I think was going on, and he said to me, "Robert, we described this in 1997." He says, "Oh yeah, this is the substrate carboxylate ring nitrogen could potentially serve as active site general base, which activates a water molecule in the deacylation step." Okay. Then Brian Soiket describes something very similar in AMC, where he's trying to make the distinction between, a conjugate-based mechanism versus a substrate-assisted catalysis. And, you know, those are really fancy chemistry terms, but they're really very, very simple. Here, you have this tyrosine over here, right? And the tyrosine, by participating in a particular type of reaction, helps accelerate it forward. Here, the tyrosine is driving the reaction. Here, the molecule is driving the reaction. So the substrate itself in the active site is pushing the catalysis of that substrate off. So it's a different idea. You have the base, the general base, the tyrosine at 150, or it could be, another enzyme, another number. But the hydroxyl group is providing the electrons. The oxygen there is providing the electrons, and this, you just go boom, boom, and you go off, as opposed to here where the driver is actually the substrate. So I went back and looked at, Chris Schofield's structure, went back and looked at Foucault Vandenacker's structure, and I says, how could you get close to that? How could this happen in KPC? And it occurred to me, and I could be dead wrong, that this nitrogen is located pretty close to that deacylating water in the crystal structure. And, you speculated that aminothiazole ring could be what could aid in substrate-assisted deacylation. I know I'm talking like a chemist, but it's being driven by the molecule. The bottom line is that it's being driven by the molecule and activating that deacylation water molecule. So, Foucault had a very good idea about this, and so I went and I sought the assistance of a computational chemist who uses accelerated rare event sampling, well-tempered metadynamic simulations. Now, if you ever said that at a bar, somebody may actually hit you. But anyway, Shozeb, very, very sophisticated dynamics, looking at the different energy wells, looking at the different relationships of the amino acids in the active site, came up with supporting the idea that substrate-assisted catalysis was actually probably going on in KPC. So what do you do? What do you do? This, okay, that's it. How do you prove it? Well, I know I'm not a chemist, but I realized that looking at some cephalosporin molecules, moxilactam looks very, very different than ceftazidime. Moxilactam is an oxycephin. It doesn't have the right electrons in the right aminothiazole ring to push that reaction forward. So I created a crazy hypothesis. Well, if you compare the activity of ceftazidime to the activity of moxilactam in every one of these mutants that we make a super TAZ-AVI resistant, the ceftazidime MIC should go up, the moxilactam MIC should not go up as much. And for D179Y, as an example, you go up from the wild type to 0.54, you go up to 64. The moxilactam mutant goes down from 8 to 2. So maybe that aminothiazole actually has some, and maybe that hypothesis is right. And when Shozeb did further covalent docking and further molecular analysis using ICM Molsoft, he was able to show that aminothiazole drives that, and that N170, another amino acid, is also important in stabilizing the chemistry in the active site. But there's more to worry about than TAZ-AVI. TAZ-AVI resistant mutants, are popping up, and we're going to be talking about this at ASM Microbe. KPC81, omega loop changes. The 243, KPC14 has amino acid changes in a different region than the omega loop, and you still get TAZ-AVI resistant. These KPCs have a different mutation. They have an insertion from 266 to 265. And, one of the good things, about having, this was a really talented graduate student that came to our lab from Pablo Power's group, Flor Brunetti, is that, you can actually hypothesize. You can make some hypotheses as, is this substrate-assisted catalysis? Is this increased hydrolytic rate for one reason? And is this beta-lactam trapping? It's really fun to think about science in a theoretical sense, because it's more than just measurements. It's the intellectual satisfaction of seeing to the other side and coming up with that. So, we also looked at imipenem relibactam. Imipenem relibactam is supposed to be used against these ceftazidime avibactam resistance. But the minute I saw this data, I called up Merck. I said, guys, relibactam doesn't get on the way it's supposed to when we did this with mass spec. So it's slower to, the D1 is slower to deacylate imipenem and ceftazidime, but it's slower to be acylated by relibactam. I don't know that imipenem relibactam resistance is going to emerge in this family of enzymes, but I know the machinery ain't right. And I know that the relibactam is not getting on the way it should be. So I think we have some things that we need to worry about. And then resistance to imipenem relibactam, if you lose the porin channels by which it goes in, you also get significant resistance. But efflux does not have an impact. How about Merovaber? Now, Merovaber, if you look at the structure, it's a cyclic boronate. The boron is part of a circle. That's why it's cyclic. And this was actually, I remember when this paper in JMed Chem by Scott Hecker was published, I was, at a Ki of 0.07 versus KPC, very good Ki. This was this was a big jump forward. I think the compounds were really very effective, and I was really pleased. And unfortunately, resistance to Merovaber occurred because of porin channels. The more porin channel mutations you have, the greater resistance to Merovaber. And that was a bit of a disappointment. We just submitted a paper to Open Form Infectious Diseases, and where we looked at Merovaber resistance in KPC producing Klebsiella. It's associated with particular repeat in the AMK36 protein and also increased plasmid copy number. So, it's not a enzymatic reason. It's more a biological, E. coli reason. A higher copy number of plasmids, more beta-lactamase, and then you decrease entry into the cell. What about ceftolozane tazobactam? When you look at ceftolozane, it's a big molecule. It's a beast. It's a beast, and it's got a lot of nice features to it that you can look at. And I love this picture because they've dissected every part of the molecule in terms of what it does. But the real business end that's got nobody explains what it is because it actually does the business is the beta-lactam bond. But every other part, helps it get into Pseudomonas, helps it stable against gram-negative, enhances anti-Pseudomonal activity, etc. So it's, it was actually put together very well. And it's shown in a lot of clinical trials that was non-superior or superior to its comparators with a very good success rate against carbapenem-resistant Pseudomonas aeruginosa, 74%. But, beta-lactamases don't read books. And they don't care what you think, they care how they can evolve. And by a series of mutations, you can develop ceftolozane tazobactam resistance. All these mutations are in the PDC or the cephalosporinase of Pseudomonas aeruginosa. They're also accompanied by other types of mutations, but these are the main drivers. So the beta-lactamases are evolving quickly. And, there has not been a real crystal structure. We're hoping to have one crystal structure out in collaboration with our partners from Venatorex very soon on the PDC88. That's being written up right now. But what we did find was that one of the amino acid mutations, and it was only by doing a very sophisticated conformational free energy analysis using metadynamic simulations. But the bottom line is that because of the substitution, the amino acid actually vibrates in the chain. So no longer being rigid, by having this sort of conformational flexibility, it can take that big beast molecule and it fits in very, very nicely. So there are other beta-lactamase inhibitors on the horizon. Venadarex is coming out with a new beta-lactamase inhibitor called taniborbactam that's being paired with cefepime. And that was actually a very, very smart choice, because when you look at the structure of cefepime, cefepime is a zwitterionic compound. This carboxylate loses its proton, and this nitrogen has a positive charge. So by being a zwitterion, it enters gram-negative bacteria like that, just like imipenem does. So that's an important feature in deciding how a partner antibiotic should be picked. Then there's a very nice series of papers written by the team at Venadarex, Dr. Chris Burns. I know Chris for many, many, many years, and I send my students in the lab and the postdocs to read these papers, just like the Weber-Bactam paper and just like the QPX paper that was published because it's really important to see the science or why the molecule was put together. The construction of the polar group, the hydrophobic moiety, what attacks to the serine beta-lactamase, and what was the proposed zinc binding, because what taniborbactam brings to the table that the other molecules that neither Weber-Bactam, Avibactam, or Relibactam bring is that taniborbactam is able to inhibit metallo-beta-lactamases. And these are some of the, you could see very good microbiology when it's been tested against a variety of strains, whether it's Enterobacteriaceae or Pseudomonas. There's a favorable profile. Chris Schofield and Oxford did some very nice structural studies on the interactions of taniborbactam with the metallo-beta-lactamases. And one of the nice phrases that he uses, this is a high energy intermediate analog approach. What does that mean? Well, when you look at the structures here, it's very interesting. Here's the taniborbactam molecule by itself, and you can see it here in the active site of NDM1. And I like to start with this picture, the bottom figure, because I could understand this the figure a lot better. So when you look at here, okay, you have your two bicyclic rings, and then this is not a ring that's closed. But look here, you have a ring that's closed. And this oxygen puts itself in a very nice position, and this oxygen puts itself in a very nice position, and this oxygen puts itself in a very nice position with those catalytic zinc molecules. Now, this means more to Albert than it does to me, because Albert's a crystallographer. But when you get that kind of coordination, it's not hard to figure out that metal is not going anywhere. That metal is not going anywhere, and you're not going to be able to coordinate that water molecule when you're all knotted up with other water-like mimics. So it's a very, very beautiful crystal structure. Now, in shameless self-promotion, we've also been interested in the boronic acid transition state inhibitors. We were very fortunate to have two compounds that we've been working with, SO2030 and MBO76, and these are the brainchilds of Emilia Caselli and Fabio Prati. They've been in this a long time, and we were able to show that at least one of them, MBO76, is stable in plasma. It's got very good kinetic activity against the particularly the Acinetobacter-derived cephalosporinase, in addition to its activity against KPC and CTXM, which we showed there. But the most exciting part of the data for us is that when you take MBO76, which is this structure, and you treat KPC, the MIC goes from 32 down to 0.06. When you pair it with cefepime in a mouse model of bloodstream infection, this is the mice that are cefepime alone treated, and in a combination of cefepime and MBO76, you get about an 80% survival. And we have plans to make the oral derivatives of these compounds, MBO76, and the most recent data we got was in a thigh infection model in pairing with cefepime, we were able to reduce the CFUs in the thigh. I don't think we're ready for prime time yet. We still have a long way to go, but it was encouraging that, using the scaffold of boronic acid, we were also able to, go forward with the research project that was successful. Other work we did in our lab before Solder, we worked with this compound ETX2514, and the the chemistry and the biology that were illustrated in this one paper in MBio formed the basis of the ATTACK trial that showed that Solder could be a very important role in the treatment of Acinetobacter infection. And FOCO got a very nice crystal structure, of there in Durolobactam in the OXA enzyme at that point. We were able to show the chemistry, by enzymatic inactivation of the cephalosporinase and the oxybeta-lactamase, so that they were good, and very good MICs were shown by that. Another compound that we've had the good success to work, this is now cefepime and N-metazobactam. But when we worked with it, it was AAI 101, and I used to say, ay. But anyway, very good. This is a derivative of, if you look at tazobactam, the difference between tazobactam and N-metazobactam is a methyl group that's added to this triazole ring. And it's really interesting, and this is why there's a big difference between what happens in the clinical world and what happens in the microbiological world, a big difference. But when you look at the details, the microbiological world informs the clinical world. And it was no surprise to me that in the meropenem study, that in the Merino study, that when they used piperacillin tazobactam versus imipenem to treat ESBLs, Pip-Tazo didn't work as well as meropenem did. And when we did this study, even though we didn't ask that question, we looked at a lot of SHV ESBLs, and piperacillin tazobactam did not work as well as meropenem. And I paused at that time and says, well, maybe, but beta-lactamase inhibitors live and die at the bench, and if you do your homework right, you'll get those things out. And another very interesting thing about N-metazobactam is that as a beta-lactamase inhibitor, it bridges all the way across the active site, so it forms like it gets on and it closes, and the formation of that kind of cross-link between lysine 73 and serine 70 is I think is an alternative mechanism for beta-lactamase inhibition. Silly enough, I went back and we looked at our data with Sulbactam, which was, the grandfather of, grandmother or whichever, and Sulbactam actually did the same thing with SHV, except we didn't realize it 30 years ago. Anyway, we've worked with this other compound, cefepime zidibactam, which is a DBO, and it's very good activity against Pseudomonas aeruginosa. And it's very good against some really, really complex strains. Because of the zidibactam component, its effect, zidibactam has an effect all on its own by being a penicillin binding protein inhibitor. And one of the last things I'd like to close with is that this is an actual true story. On a Friday night, I was called by Dr. Shimamura at Ohio State, and she had a child that had a renal transplant. And this kid with the renal transplant was bacteremic with this Stenotrophomonas for almost 60 days, on and off bacteremia. The graft was a true risk. So she called up, and Maria and I talked about it. Let's come up with a combination that could overcome this bad Stenotrophomonas infection. So we, based on what we knew biochemically, that metallobeta-lactamases could not inhibit, Astrianam is not inactivated by metallobeta-lactamases, and knowing that the avibactam could inhibit the L2 beta-lactamases, we put the combination Tazavi and Astrianam together. You see this nice synergistic disc. We ended up saving the allograft. I was just called a couple of months ago where Dr. Shimamura had a follow-up with that renal transplant patient. The renal transplant patient is still alive, still has the renal allograft, and is no longer bacteremic. But we got rid of the bacteremia, and it was probably a result of this calcification in one of the pulmonic valves, pulmonic artery, that served as an endocarditis equivalent. Then we went on to show in an animal model that it also worked. And my last thing is that we had a discussion today about mycobacterium and using creative approaches to mycobacterium using double beta lactams. And one of the new compounds we worked with is durolobactam, and the ability of durolobactam to work synergistically. We think this notion of target redundancy, where you can use multiple agents that hit multiple penicillin binding proteins, LD transpeptidases, DD carboxypeptidases, in the mycobacterial cell wall, may be a very good adjunctive therapy to our current treatments. So what do we say now? Well, it's going to be hard to know how to use these beta-lactam, beta-lactamase inhibitors. They're not inferior to our standard therapy. They're quite superior. But, their real impact is going to be on these multi-drug resistant organisms. And I think multi-drug resistant Pseudomonas and CRE are the areas where these ought to be used. I think if they're used against first-generation infections, we're throwing away a very good tool. And the clinical microbiology lab is going to be very challenged because how to best test them. It's going to be very hard for them to know. And how many beta-lactamase inhibitors will they test for you? I told you about 11, and you see how complicated it could be. And we still have a lot of challenges ahead. We have to figure out how to use these agents better. I'm looking at better structural biology techniques to inform us, as well as analytical tools, cryo-EM, atomic force microscopy, etc. And, one of my most favorite books was by Peter Piot. He was head of the, he discovered the Ebola Marburg virus, and he always said that we had no time to lose. And I just edited his thing, A Life in Pursuit of Deadly Viruses. I have a life in pursuit of these deadly beta-lactamases, etc. So I want to thank our sponsors for this talk, the Faculty of Medicine. Oh, okay, you do it, Al.
First. Thank you very much, Robert. This was quite the whirlwind through the beta-lactamase field and the beta-lactamase. You're sitting very comfortable, but that doesn't mean there are no questions. There might be questions in the audience. There's a microphone. So if you have a question, that way the people online can also hear it.
I want to acknowledge Dr. Paul Roy, who's sitting in the audience. Dr. Roy, he's sitting. He was listening in. Dr. Paul Roy. He and I had many very fruitful conversations years ago. He's a great scientist.
Oh, thanks, Robert. That was a terrific talk.
Okay. He indeed listened. So you can come back up here because Sebastian has a question.
Thank you for a great talk. This is really inspiring. So, a simple question. So, do you think there's really no chance for it? I saw that there are single agents that are both antibiotics and beta-lactam. Do you believe a single agent will ever be that useful, or we're going to have to go with multiple combinations?
I think about that a lot because I know what the current paradigms are. We want to use one drug for one bug, and we want to do it once. I think the evolving landscape of multi-drug resistance is not going to be able for us to entertain that strategy. There's some other bugs that are even more complicated, and they have so many different mechanisms of resistance that evolve. And the one thing we really haven't had a good We don't understand the stress response and mutational frequency. We haven't gotten ahead of evolution. Evolution is far ahead of us, and we can't anticipate the next steps. And I think combination, and I know I'm not in the majority of people, but I think for some of these really difficult to treat infections, combination drugs are going to be the way to go, because it's going to be hard to overcome one mechanism only.
And second question, so I did not look at the structure of your inhibitors, like the MB.
Oh, so yeah.
So is it amenable to also make it Zwitter ionic?
That
Something that having one methyl and having a
I'll ask my chemist. I don't know if it can. You know, basically it's triazole. You know, the delivery of that is the triazole there, because it forms interactions that really improve its affinity. It's a good question. Maybe we should make it into a Zwitter ionic compound, because it works really well with cefepime. Even though the MIC is not as good with cefepime, the biological properties of cefepime in an animal model are much better than meropenem. Meropenem is passed through the kidney too fast, so we don't look as good.
Thank you very much. I'm a clinician, so I didn't really get a lot of the chemistry. Apologies.
Well, neither did I, so it's okay.
So, but I really, I was wondering, most of the backbones, the beta-lactam backbone, are those with anti-pseudomonal activity. But in the clinical world, there are kind of two different groups of patients: the ones that come in with sepsis due to an enterobacterialis versus, you know, those that have a pseudomonal sepsis. So would it not make sense to kind of, as an approach, have two different pathways, a beta-lactamase inhibitor that's geared towards E. coli and a smaller group that is geared with anti-pseudomonal?
Sulbactam, Darilobactam is that kind of paradigm drug in my mind. In my mind, it's an anti-Acinetobacter drug. I would not use it against any other bug other than Acinetobacter baumannii. And the reason being is that the clinical trial was specifically designed. The biochemistry of Darilobactam actually helps us there. And the sulbactam really is very important when it comes to Acinetobacter because of its PBB effects. So I think some compounds will be developed as niche drugs. There's going to be one indication. They're going for one type of bug, and I hope they make their financial return on it, you know, because but I think the government is going to have to help them. So there's going to be different financial incentives for these niche drugs, I think.
But how about a drug that would be really for an E. coli that's an ESBL? Because that's the majority that we see in the hospital. And we don't actually want the anti-pseudomonal activity for 80% of the cases. And would that be possible?
I think a drug like cefepime and tazobactam that just got over the finish line, that's a tazobactam derivative that's done very well with E. coli, UTI. I think cefepime taniborbactam also did very well with E. coli, UTI in the RECOVER trial. So I think some of them may be useful. I think some compounds are still in development that are going to be specifically for E. coli and Klebsiella ESBL producing organisms. I think there may be some still in development, although I can't say to that.
Well, most stimulating presentation. I'm going to ask you to speculate about some of the structural considerations in designing these drugs, because a 6-month to 2-month layover for developing antimicrobial resistance, it's a little bit disheartening. I was wondering whether a way to model structurally, approach to modeling the structure might be to find a way to destabilize some of the coordinate bonds. And I think that this can be done by looking at some of this metadynamics from the perspective of entropic considerations. Is there a potential to open up this window, especially when one's considering adjuvant therapies, to find ways to destabilize those coordinate bonds?
There are programs that are being developed right now, and my good collaborator, Dr. Shozeb Haider at UC London, he's developing programs that look at specific amino acid sequences, and by categorizing these OXA enzymes that he's looking at, he's trying to be able to predict how their phenotypic and how their biochemical behavior is going to be based on the amino acid sequence. So it's a big project. It's a lot of computer power, and we have to do a lot of crystallography and a lot of MIC testing, but he's trying to be able to do that. So I think people are heading in that direction to be able to look from an amino acid sequence and translate that into a phenotype and into a biochemical type. So I think people are headed to that. I think the field is still in its infancy. We don't know enough yet that we can do it. We know how certain families of enzymes behave, and we know what the hotspots are, but we've been dealing with those enzymes since 1964. So we have, a lot of time, to see their evolution. Some of these enzymes that we've only studied them for 10, 20 years. We don't have not enough time yet.
How long will it take to develop that program, and by how much can we expand the how do you say, the validity or the value of those particular?
I hope he develops it soon. The analysis soon. I think it'll take about 2 to 3 years to be able to test all the different things. It takes time. It's not something that can be done overnight, in my opinion.
Final question.
Dr. Bonomo, thanks very much for the talk and for your continuing contributions to the field. Could you maybe just provide your perspective on a standalone BLI, whether you think that companies or consortia should be developing beta-lactamase inhibitors, perhaps novel inhibitors, to be paired at the clinician's choice with a beta-lactam of their choice?
It's potentially a very good idea because you can personalize your approach. I think it's intellectually interesting. It's intellectually inviting. The problem is the reality. The reality of having a standalone beta-lactamase inhibitor has with it many assumptions. The first assumption is I know who to partner it with. And that's because each one of these has a little bit different PK, PD, a little bit different behavior. So I got to be able to pick the right partner for that beta-lactamase inhibitor. My lab has got to be able to test to make sure that that beta-lactamase inhibitor that I pair with the beta-lactam I want is going to be as potent as the others. It will also be extremely difficult to do a clinical trial, saying, well, this combination works better than that one because I picked it on the fly. I think few people can do it because they know the drugs well enough. I'm sure there are people out there that can say, I know exactly what to pair with what to get the effect I want because I've been studying these damn things for 25 years. But that's not the level that you're going to have in a surgical intensive care unit at 2 o'clock in the morning. That's not the same level you're going to have in an emergency room. That kind of decision-making process is going to have to be further on down the line where it's people really know what they're doing that can help you. But I think an empiric, let me pick what I want with this, is not going to happen.
I would like to thank you again for really stimulating, exciting.
Thank you for inviting me.